My name is Mark Ramsdale. I'm the head of training at the MRC Centre for Medical Mycology. I'm been a fungal biologist for about 30 years and a long-standing member of the British Mycological Society. So it gives me great pleasure this evening to introduce Sandra. Sandra is an associate professor at the University of Lincoln. She's an evolutionary ecologist and is interested in understanding how plants interact with their environment. Sandra is particularly interested in the effects of climate change on plant-soil interactions and how soil microbes can contribute to sustainable food production and net zero calculations. So please welcome Sandra, and she's going to talk to us on the potential of arbuscular mycorrhizal fungi and cyanobacteria for sustainable agriculture. So over to Sandra.
Thank you, Mark. Thank you. And good evening, everybody. I cannot see you, but hopefully you are on the other side. First of all, thank you very much to the BMS for this opportunity. I did got some funding that I'm going to present a little bit of the research that I've done thanks to that funding. And it's, of course, a pleasure always to share knowledge. So the title of my presentation is here, and what I am going to be talking in the next minutes is about, in my opinion, the huge potential of these very pretty organisms, arbuscular mycorrhizal fungi and cyanobacteria for sustainable agriculture. So this is a little bit my plan for today. I am going to start this talk by introducing a little bit about myself. Where do I come from, basically? If you might be wondering this weird accent, you will get the answer of that as well. And how did I end up at the University of Lincoln, where I am working with these amazing soil organisms? Then I'll give you a little bit of an overview of the history of agriculture, where are we now as humans, and a little bit of thoughts or food for thought of where things might be heading, in my opinion. And this is quite my personal opinion as well. Then we will look at the potential of these arbuscular mycorrhizal fungi. What are they? What do they do? A little bit on cyanobacteria as well, to then jump into what do we know when we inoculate plants, crops, with both types of organisms, both arbuscular mycorrhizal fungi and cyanobacteria. At the end, there is going to be time for questions, as Mark was saying, so please use the chat. I would be very, very happy to try to answer and discuss these things with you. So let us start a little bit from the beginning. What do I do? And what I would like to do is try to put a label on myself, which I always find really, really difficult. As Mark said, I guess I'm an evolutionary ecologist. What I'm really interested is in plants and how plants interact with soils and with environments. I teach at the University of Lincoln as well. I do some research, and I always tell my students, if there is a plant, probably I'm next to it. And if there is soil, I am just there looking into it as well. I assume that normally for this type of audiences, I don't need to tell you much to convince you how important soils are and soil functions to humankind. This is illustrated in this diagram, but soils provide crucial ecosystem services that actually enable our life on this planet. You can think about functions such as obtaining clean water, soil contamination reduction, help in regulated climate, nutrient cycling, carbon sequestration, you name it. And of course, soil provides most of the food and energy we consume as humans. Thinking about food, of course, it is estimated that 95% of all our food comes directly or indirectly from soils. And therefore, I think that understanding this precious resource and conserve it as well, it is crucial. And that's why I think I started being interested, probably when I was a child, in these plant-soil interactions. What is going on? But what is soil? For many years, people thought about the relationship between plants and soil as being very complex. And I'm talking about the good old age. So plants were growing, something was happening in the soil, but we really didn't know what was going on and how. And for many years, the soil was actually referred as a black box. It was an opaque system. You couldn't see something was happening. Plants were happy or not, but nobody knew what was happening inside and how. The good thing is that quite rapidly, people realized that a healthy soil and thus a healthy crop was depending on the physical properties of soils and also the chemical properties of soils. And by this, I mean things like the amount of nutrients that the soil contains, the amount of organic matter, the structure of the soils, whether they are really sandy, whether they are more clay, how fast the water can infiltrate in the soils, and so on. And of course, also the soil pH, salinity, many other factors I'm not mentioning here. But now, a bit later on, this black box opened in the sense that we are starting understanding more and more what was happening below our feet, inside the soil, how was everything getting placed. And we began to understand that not only the soil physical and chemical properties are important for plants, but also, and quite crucially, I would say, their inhabitants. Or in other words, the biology of the soil. And in here, hopefully, you can see lots and lots of different organisms, which of course it's not all of them, from millipedes, from bacteria cells, fungi, of course, mites, nematodes. One of my favorites actually the tardigrades on the right-hand side, and collembola, for example. All these inhabitants are very, very important for soil ecosystem processes and functions. So from the smallest to the largest, soil contains billions of bacteria cells. And of course, in terms of abundance, it has been estimated that there are more soil microorganisms, and I'm just talking about the little organisms in soil, in a teaspoon of healthy soil, and that is important, than there are people on Earth. So there is a lot of things happening in the soil, as you can imagine. And this is one of the interesting things, I would say. Even though we do know that soils are crucial and that we as humans depend on this resource, it is estimated that 1/3 of all the soils in the world are degraded. That's 1/3. That's a lot of soil. What are the degradation threats affecting these soils? You can see them on the upper left-hand side. These degradation threats may include, depending on the place we are talking, factors such as soil sealing. So when we build on soils, we are decreasing the amount of air, we are changing all the nutrient cycles, and so on. Factors like contamination, acidification, salinization, nutrient imbalance, and also loss of soil organic matter and loss of biodiversity. And this is quite crucial, I would say. All of these factors have then consequences for food production and ecosystem degradation. And what leads, and this is also the worrying part, I would say, to more social inequalities and poverty, for example, where the food is produced, how it is produced. And at the end of the day, of course, we are sharing the same planet. So that's my point about why we should care about soils. So this is precisely where I'm trying to make some meaningful differences. This is just a snapshot of one of my current projects that is looking into working towards this net zero potato supply chain. This project has been funded by Innovate UK, and we have collaborations with Branston, Beehive, and among other industrial partners, as you can see at the lower part of this slide. And what we have been doing for almost 3 years now is testing, among many other things, how different microbial inoculations, how different types of fertilizers, and how different innovative cultivation techniques can be employed to reduce not only soil greenhouse gas emission, which is contributing to climate change, CO2, methane, nitrogen, NO2 gases, for example, but also the emissions related to transporting and storing these potatoes. So not just potatoes today. As I mentioned before at the start of the talk, I would like to mention here a small research project that was funded by the British Mycological Society, where I investigated whether the dual inoculation or the dual application of arbuscular mycorrhizal fungi and cyanobacteria can improve wheat growth and yield. So this is something I'm not presenting now. I'm going to talk about a bit later on. So this is a little bit of what do I do, but I don't know if you agree with me. I always find it fascinating to ask people, how did you actually get in here? Why are you doing this in here and now? This is a young version of me in the left-hand side. I am coming from a farming background, from a little village somewhere in Catalonia. And I have a great passion for especially mushroom foraging, as you can see here with some of my family pictures. It's a big activity. But how did I get here? I suppose my academic journey started many years ago in sunny Catalonia. You have it in here at the end of the map. I was born in Manresa, a city very beautiful for the cathedral and many other places as well. And then I did my undergraduate degree in plant biology at the Universitat Autònoma de Barcelona. After that, I decided to go a bit north, and I went all the way up to Oulu in Finland, where I did my PhD. I didn't take a car, I took a plane actually. And in my PhD, what I was doing was looking into whether the interaction between arbuscular mycorrhizal fungi, which I'm going to be talking about a bit later, is dependent on the gender of the plants they colonize. So some plant species have separate sexes, as humans, for example, females and males in the case of plants, females and hermaphrodites, and all sort of breeding systems as well. And I investigated whether the relationship that these plants with different genders have with fungi depends on the gender of the plants. And I can tell you, it does. I was loving Finland so much, I would say that I decided to stay after getting my PhD. And I started moving a bit towards the south already. And I went to Jyväskylä in central Finland for a couple of postdoctoral positions, where I basically continue studying arbuscular mycorrhizal fungi. So all in all, I think I've been with arbuscular mycorrhizal fungi for quite a lot of years. After finishing my postdoc, I was very lucky to get a Marie Curie fellowship, and I moved to the University of Lincoln, where I am currently. I spent a couple of years as a Marie Curie fellow, still with mycorrhizal fungi, and then I was quite lucky to get a lecturer position, a senior lecturer position, and recently, a couple of years ago, I was promoted to associate professor. Had a maternity leave in there, which I think is worth to mention as well. But here I am talking about fungi to you from the University of Lincoln. However, enough about myself. And what I would like to do in the next slides is to give you a very brief history of agriculture. Throughout our history, humans have been looking for solutions to obtain one of the most basic necessities, food. Agriculture actually began in an independent way in different parts of the globe, more or less at the same time, it is believed, with at least 11 separate regions being involved in these origins of agriculture. So by agriculture, of course, I refer to the fact that people started understanding more plants and planting plants and getting crops. The development of agriculture about 12,000 years ago changed the way that humans lived. They switched from a more nomadic hunter-gatherer lifestyle to more permanent settlements and farming systems, which of course was one of the bases of our societies, I would say. The invention of agriculture increased food production and led to a rapid growth in the human population around the globe, which reached more or less 50 million people by the Bronze Age. All of this, I guess, sounds very positive, looks quite pleasant from these pictures, if you like, but throughout our history, advances in agriculture, which have been fundamental for our society, have been going hand in hand with an increasing number of people in our planet, and also an increase in atmospheric CO2. As you can see here, and in the next slides, this will be, or is still creating, of course, big problems for our current and future societies. The increase in atmospheric CO2, climate change, more extreme temperatures, more weather extremes, and so on. Jumping quite a lot of years, as you can see, and more or less by 1760, when the Industrial Revolution began in Britain, the world population already reached around 600 million people. At this time, there was an accelerated farmland expansion. Lots of more farmlands were created, and more innovative applications of technologies to food productions were achieved, which of course is also very, very positive. A bit later on, around 1950s or so, there was what we call the third revolution, the third agricultural revolution, or the Green Revolution, which saw an increase in food production through the use of these high-yielding cereal varieties. A lot of research and effort going on in there, with higher rates of fertilizers, synthetic agrochemicals, and controlled water supplies. By 1950, the world population was 3,700 million people, and the CO2 concentration passed the 310 parts per million. So a huge increase in here, as you can see. Where are we now? Well, we seem to be facing the fourth agricultural revolution, according to some people, under a world population that has already passed the 8,000 million people, and an atmospheric CO2 concentration that has reached almost 430 parts per million. In one way, of course, this fourth revolution is revolutionary, as the word says, because the introduction of artificial intelligence, for example, and more power autonomous robots, this is helping us to produce food, more food, but at what price? And here is what I would like you to stop for a second and think. How do you think our planet will look if we continue with these practices, or if we change the practices on the other hand? Which kind of options will we have as a society if the world population is expected to reach the number you see in the screen, more or less by 2100? What are we going to do with such amount of atmospheric CO2? What are we doing with the climate change and so on? Some people claim or think that maybe we could start thinking about producing agriculture in other planets. I'll leave it in there. However, what I would like you to show is that I think that part of the solution is actually to look into sustainable ways to produce food. And for that, I believe we need to get a step back and start looking back again into the soil to really understand which organisms live there, what do they do, and what can they do for us. And on the other hand, what are we doing to the soil organisms as well? This black box that I was referring to in the beginning of the talk is where I think at least part of the answers might be. So in the rest of this talk, I'm going to be focusing on arbuscular mycorrhizal fungi and cyanobacteria, but please do not forget that there are lots and lots of other soil organisms and many other types of potential bioinoculants that could be also very useful for sustainable agriculture. So let me start with my favorites. I would say arbuscular mycorrhizal fungi. What are they? Some of you may have heard of them, some of you may not, but mycorrhizal fungi, or mycorrhizas, the association in the root, are mutualistic associations between some soil fungi and the roots of plants. It is known that around 70% of all land plant species do associate with these important symbionts in their roots. So I always tell students, like, if you go outside, if you now go outside after this talk and you look at the plants around you, if you take one plant by random, most probably will have mycorrhizal fungi in their roots. This is how they look like. In this picture, you can see some fungal spores in here, and you can see the roots of Plantago lanceolata, plantain. And in this association, how it works, basically, the plant allocates photosynthates, carbohydrates, sugars, to the fungus in exchange for mineral nutrients from the soil and water. So normally, this is a mutualistic relationship, and mutualistic means that both partners benefit from associating with each other. I give you sugar, you give me nutrients, it's a win-win situation. And for the fungus, they cannot live without a plant. This type of arbuscular mycorrhizal fungi are biotrophic fungi. They are fully depending on the hosts for their carbon. They cannot photosynthesize and they cannot decompose, for example. From the plant point of view, it's usually very beneficial to associate with this fungi. Not always, as we will see now. But of course, plants can survive without arbuscular mycorrhizal fungi. And I'm talking about arbuscular mycorrhizal fungi because some plants develop an interaction with other types of mycorrhizal fungi. So how does this work? As you can see from this slide, arbuscular fungal spores germinate in the soil when the conditions are favorable for them and start producing hyphae that try to find the host. So for the fungus, because they're fully depending on the plant, it's very important that they make sure that there is a potential good host. So there is a lot of chemical communication between the plant and the fungus happening in the soil. When the hyphae detect a host, they produce this structure, this hypopodium, and it's a bit of an enlargement of this hyphae, and they start penetrating the plant tissues inside, producing internal hyphae, as you can see from here. These external hyphae in the soil, what they do is they enlarge the available surface for nutrient uptake for the plants, which is of course very important for some nutrients, such as phosphorus, where its movement is quite limited in the soil, as we will see in a second. On top of that, having mycorrhizal hyphae instead of producing roots to take these nutrients and this water is usually much more cheaper than producing the roots itself, because the hyphae are much, much thinner compared to the roots. For the fungus, as I was mentioning, the symbiosis is always beneficial, as arbuscular mycorrhizal fungi are obligate biotrophs and they are fully depending on the hosts. However, not all hosts are good or equally good. The relationship to the plant, as I said, is usually beneficial, as this increases the supply of nutrients and water, but it's also costly in terms of carbon. There's a bit of a discussion still going on of how much carbon can actually the plants allocate to the fungal partner, but some estimates are available that they say that between 4 and 20% of the total photosynthetes are actually allocated to the fungus, and that's quite a lot of resources. The nutrient exchange take place in structures called arbuscules. You have a little diagram in here, which comes from the Latin little trees, simply because the hyphae penetrate inside the plant cells. They start ramifying a little bit like a little tree, if you like, which increases then the surface area for the nutrient exchange between the two symbionts. As I mentioned, though, the relationship between plants and arbuscular mycorrhizal fungi is usually considered to be mutualistic, so both partners benefit from this, but this is not always the true outcome, I would say. The outcome of this relationship at the end of the day ranges from being mutualistic, both partners benefit, from being parasitic, so one partner is benefiting and the other is getting a detriment, depending on both the abiotic conditions, environmental conditions, as we will see in a second, amount of nutrients, for example, but also pH, pollution, and many other factors, and also the biotic context, so who else is in there. As you can see, or what I'm trying to show you in this graph, on the left-hand side, we have a plant root without mycorrhizal fungi. So the plant is able to take all the phosphorus that in this case is really immobile in the soil by using these radicular hairs in here. When this phosphorus has been absorbed, there is a depletion zone. So there is no more phosphorus that the plant can access, because this phosphorus in here is very, very slowly moving towards the root. However, using arbuscular mycorrhizal fungi and with the production of this external hyphae, the plant is able to enlarge the amount of phosphorus that is available for them. This is very, very beneficial, as you can see. However, as I said, it's not always beneficial. And maybe, as you can imagine, if there is a lot of phosphorus, why should I give sugar to a fungus? Why should I not just take the phosphorus myself? And this is what it has been known for many years already. This mycorrhizal benefit depends very strongly on the amount of phosphorus available in the soil. If there is not that much phosphorus, if phosphorus is limiting, having mycorrhizal fungi is beneficial, as shown in this graph. So this is showing you the plant relative growth rate, how much they grow per day, with mycorrhizal fungi in gray and without mycorrhizal fungi in white. So when there is relatively low amount of phosphorus, there is a mycorrhizal benefit. It is good for the plant to invest in this symbiont to get the phosphorus. However, when the phosphorus concentration in the soil increases, it doesn't really matter. It's costly to have this symbiont. So with this in mind, do you think that arbuscular mycorrhizal fungi can be beneficial for agriculture? The answer is yes, but with some comments to add, I would say. We do know that most plants usually benefit from mycorrhizal fungi, not only because of this enhanced nutrient uptake I was talking about, but also because arbuscular mycorrhizal fungi can also help improving soil stability, for example. With the production of this external hyphae, with the production of glycoproteins, the fungus is helping the soil particles to glue a bit together, which is preventing a little bit erosion, for example, which might be quite beneficial in certain habitats. But mainly through the increased nutrient uptake of mainly phosphorus, but also nitrogen and other micro and micronutrients, plants inoculated, colonized by arbuscular mycorrhizal fungi are normally better protected, bigger, and protected against pathogens and herbivores. So the answer is yes, it is a good idea. And this is just an example. There are many, many on the scientific literature, of course, of these beneficial effects. So this figure is showing the results of a meta-analysis where the authors look into whether the effects of adding arbuscular mycorrhizal fungi were beneficial or not, looking into several cereal plants. They searched the literature and they could actually include 168 separate studies that look at the addition of mycorrhizal fungi compared to a control condition without inoculation of fungi. And what they do, or what we do when we do this meta-analysis, is calculate something that is called an effect size. The way to interpret this graph is basically looking into whether the black dots are touching this horizontal zero line of the effect size. So anything that is above this zero, it means that mycorrhizal fungi have a beneficial effect on the plants. Anything that is touching the line, or if it would be below the line, it would mean either no effect if it touches the line, or that the addition of mycorrhizal fungi could actually be detrimental for the plants. So as you can see from here, authors show from these 168 studies that overall there was a positive arbuscular mycorrhizal fungal effect, if you look at this first dot in here. They calculated this effect to be a 21% increase in yield, which is quite considerable. But as shown also in this graph, this was also dependent on the crop that was studied. So for example, in barley, they did not find a mycorrhizal effect, neither positive or negative. In maize, the effect was positive, mycorrhizal fungi was good, the same for millet, rice, and wheat. Also in here, just mentioning now that sometimes, or many times actually, we see a difference between the results that we see in lab experiments, more in the control setting, if you like, compared to field trials. So in some of these cases, there is a bit of a difference depending on whether the study was done in the field or in the lab. So this is not only for cereals, and I'm just showing you now an example of an experiment or meta-analysis that we did in my lab in this Innovate UK funded project, focusing on potatoes, very important crops, of course, and very important food source. We review very similar as before. We review all the literature and also observe that the application of arbuscular mycorrhizal fungi was beneficial overall for potato crops, as you can see from this graph. So in here, basically the axes are rotated, but it's the same idea. If the black dots, the mean values of these effect sizes do not cross the zero line, is not touching the zero line and is positive, it means that the addition of mycorrhizal fungi was beneficial for potato production. And this is actually showing an increase in tuber yield, how many more potatoes, tubers the plants produce. Again, we found in this case no differences between whether the studies were done in field conditions, a bit more real, or in pot studies. But what we did see, it was a difference depending on whether this mycorrhizal inoculum came from a non-commercial source, so like a lab-based inoculum, as I would have in my lab, for example, or whether this inoculum was commercially available. Many bioinoculants can be bought and used. So this is a bit about arbuscular mycorrhizal fungi, looking good, right? But what about cyanobacteria? Cyanobacteria, you have a picture here of some of these fantastic organisms have gained quite a lot of attention in recent years because of the multiple applications in biotechnology. Cyanobacteria, they are not fungi, they are prokaryotic organisms that can photosynthesize. They do have pigments, a bit like plants basically, and are found in almost every habitat on Earth. They are very ancient, they are very abundant, and they are very important. They exist in many different morphologies, as you can see here, from being unicellular on top, like one cell or couple of cells together if they want, to being filamentous, or more like a thread. Biological applications, of course, I'm not talking about them in this talk, but may include the production of, for example, antivirals, antibacterial compounds, bioplastics. They can be actually used to produce alternative energy sources. So yet again, very much related to climate change and so on, and net zero. But importantly for this talk, some species of cyanobacteria can be used as biofertilizers. And some have the ability to fix atmospheric nitrogen and make it available for plants. And this is where the link with cyanobacteria and agriculture comes for me. So this is an example of a species of cyanobacterium. Some of them can fix nitrogen, as I said, using a specialized enzyme called nitrogenase that some of them have in these specialized cells, yet again, called heterocysts. What this enzyme does, it takes the nitrogen gas, which is very abundant, we have plenty of it, and it converts it into ammonia. And this ammonia is a form that plants and other organisms can use to build amino acids and other essential molecules. So, what do you think? Do they work in agriculture? Again, the answer seems to be yes, but it depends. So some studies report actually that this nitrogen fixation in the soils by some species of cyanobacteria can be up to 28 kilograms of nitrogen per hectare. This is quite a lot. And this number comes actually from a wheat crop. As another example, and yet again, you can find many in the scientific literature, in this study, the authors conducted an extensive analysis of the bacterial communities found in the soil surrounding the roots of cotton fields. Using metabarcoding methods, the authors characterized the microbial communities, so they look which kind of microbes they were associated or not with the roots of cotton plants, and they selected 5 native cyanobacterial strains. The names of these you can see in this graph. They just put some numbers, G23, G26, and the last one is actually a species of nostoc, which is quite well known perhaps. These 5 strains, together with nostoc, were investigated for their plant growth promoting activities using cotton plants. So they basically took these strains, cyanobacteria strains, from a field of cotton, they isolate them, and then they put them in experiment with cotton plants. And as you can see from this graph and the pictures, compared to this untreated control, so plants without cyanobacteria in the soil, some combinations, some of these strains actually improve the plant weight. This is compared to control plants. So in this case, this G23, for example, you have a picture in here, and you can see how much difference there is between the plants, had a positive effect on the aerial biomass of the cotton plants, but not the roots. In this G42, another strain that they found, the effect was positive, but was the other way around. So the plants look more or less the same. However, the roots were massively increased. Many more roots means, of course, that they might be able to get more nutrients and so on. I'm not selling them, but it seems that I'm selling cyanobacteria to you. However, also, for example, from this experiment, always remember, not every single organism is going to be positive. So in plants inoculated with this nostoc, for example, the effect was negative. So it would have been better for the plants not to have the cyanobacteria in the soil. So, with all this information in mind, what about if we actually take arbuscular mycorrhizal fungi, which are good in giving plants phosphorus, water, and other nutrients, and we also add this cyanobacteria into the pots, that they might actually give nitrogen to the plants? That would be like a win-win situation, right? As we have seen, when I started thinking about it, I found plenty of independent, separate studies looking at soil inoculation with either arbuscular mycorrhizal fungi, it has been known for many, many years, or cyanobacteria, how these organisms can improve soil structure and fertility in many different crops. However, I could not find that many studies that they evaluated the synergistic effects of dual inoculation. Would the plants win more whether they're inoculated with both organisms? And when I did that, as far as I was aware, there were no studies that did that with wheat plants. So this is what I did with funding from the British Mycological Society, which I'm thanking again, because this makes a difference for science and for many people, of course. What I did a couple of years ago already is to establish a pot experiment with 400 wheat plants using 3 different species of cyanobacteria and 3 different species of arbuscular mycorrhizal fungi. And basically, I had all the possible combinations between these 2 treatments. I had some control pots where I had wheat plants without cyanobacteria, without mycorrhizal fungi. And I had pots that were 3 different species of cyanobacteria were introduced. You have the names in here if you are curious. This Anabaena variabilis, another species of Anabaena, and also nostoc. And for the arbuscular mycorrhizal treatment, the same idea. Control pots without adding any arbuscular mycorrhizal fungi, and then pots inoculated either with this Funaliformis claroidiogleu, which I refer as CLA, Claroideoglomus tunicatum, ETU, or Funaliformis mossi. which are quite well standard arbuscular mycorrhizal species. And of course, all these organisms occur in soils. So I did this, I put pots, I put lots of seeds, I put lots of organisms in there, and I look into the germination of these plants and the growth of these plants, and as well as I took some measurements of the physiology of the plants, mainly photosynthesis, and estimated the amount of biomass produced at the end, and also the yield, which at the end of the day is what matters for a farmer, for example. So this is how they look like. They looked like, because now, of course, they are harvested this couple of years ago. So the plant established relatively well. 19th of May, you can see very nice establishment. And a month later, in June, you hopefully can see already some seeds in here being produced. So what did I find? Did it work? Yes and no, I would say. Overall, I found that inoculation with arbuscular mycorrhizal fungi did not affect the photosynthetic rate of plants. So this was just one measurement done on the amount of photosynthesis that the plants were doing. And this is presented in this graph. There is a bit of variation. So in control conditions, without any cyanobacteria, and depending on the combination of different mycorrhizal fungi, you can see that the photosynthetic rate, the amount of photosynthesis that the plants were producing, was more or less similar. And you can see some differences between some treatments, even though they were not statistically significant. But how is that translated to plant growth? Let's see. I found that inoculation, I'm going backwards, apologies, there we are. I found that some variation in the total amount of plant dry weight at the end of the experiment. Some combinations, yet again, between the cyanobacteria and the mycorrhizal fungi use looked a bit better than the others, and some look actually worse, I would say, even though in this case there was no statistically significant difference in plant growth. But as you can see from here, some combinations didn't work that well if we are assuming that more biomass will produce more yield at the end. So for control plants without a cyanobacteria, the worst was actually to have this clarideal species as well. Biomass, photosynthesis, very important, of course. But at the end of the day, what about the amount of yield? How much cereal can these plants produce at the end of the experiment? And this is what a farmer might be interested in. In this case, both arbuscular mycorrhizal and cyanobacteria influence the amount of yield produced in wheat plants, depending yet again on the combination. So hopefully you can see a theme in here. For plants without cyanobacteria in control conditions, plants that were inoculated with this mossy species were the ones that produced the highest yield. The worst, yet again, if you are a wheat plant and you don't have cyanobacteria, it's a very bad idea to also have this clarideal species of mycorrhizal fungi. So more differences in here, depending on the combination. If you were a plant inoculated with this nostoc, the worst would be to also do not have mycorrhizal fungi, so green in non-mycorrhizal conditions. And the best combination would be to have nostoc and this Etunicatum species. And a very similar result was found using this other cyanobacteria species, but not this Anabaena. So I assume this is a bit of a complicated graph, but the message here is that there is a significant interaction between the two players. Who is the cyanobacterium I'm adding? Who is the mycorrhizal fungal species that I'm adding? It really depends the outcome for the plant. So taking everything together, and if I would be in a lecture hall, I would ask you and look at the reactions, of course, but do you think that arbuscular mycorrhizal fungi and cyanobacteria can be used in sustainable agriculture? I would like to say yes, they can, but hopefully, as you can see from this presentation, we need quite a lot of more research to understand what is going on and under which specific combinations of plants, microorganisms, cyanobacteria, mycorrhizal fungi, we get the best outcome. And especially, we need more field trials under real conditions. For example, and I am in the next 2 slides, I'm mainly focusing on arbuscular mycorrhizal fungi, but I'm pretty sure it applies to almost any soil organism. It is well known that arbuscular mycorrhizal fungi are especially beneficial under certain conditions. In this case, arbuscular mycorrhizal fungi are very good when there is a low original inoculum potential. If you go to a field that they don't have that many mycorrhizal fungi, potentially it might be a good idea to reintroduce some of these species. And as we've seen before as well, it is a good idea when the amount of phosphorus in this case is not really, really high. If we have lots of nutrients, mycorrhizal fungi, they will not do anything beneficial for the crops in terms of nutrients. However, and this is the big howevers, the results seems to be very much context specific in regards to the plant fungal bacteria combination used, the strain, the conditions, and so on. Moreover, I would like to just show you an example. The positive results that we normally see in pot experiment or in more controlled lab conditions sometimes do not translate in real field conditions. But why is that? There might be many explanations, of course. The amount of nutrients present in agricultural fields might be one of the explanations in many of these cases, but also several studies have indicated that applying commercial products usually result in less prominent effects than the ones we see in the labs in more controlled conditions. As an example, as I said, in this study, the authors evaluated how good different inoculums were, depending on whether the arbuscular mycorrhizal fungi inoculum was prepared in a lab, it was more lab grown, as it says here, or whether it was a commercial product. They estimated the amount of root length colonized by fungi after a certain time, and as you can see here, the amount of hyphae was much, much higher when we were using lab-grown fungi compared to commercial products. And most importantly, I would say the amount of arbuscules, these structures where the nutrient exchange take place between the plant and the fungus, also which was much lower in the commercial products. Other studies comparing these commercial and lab-grown products have indicated that sometimes the commercial inoculums that you can buy, sometimes they actually do not contain the species that they say in the labels. Sometimes the spores are actually dead, so you are buying an inoculum that is potentially dead, or even sometimes this inoculum may contain some pathogens. And this is, of course, not the case for all the commercial products. Let me be clear with that. However, I think this highlights the need for a more comprehensive framework for basic testing guidelines and quality control in inoculum production. I'm talking about mycorrhizal fungi, but as I said, potentially this might apply to other bioinoculants as well. Finally, I've been mainly focusing on the plant's point of view and talking about sustainable food production. We want to produce more food in better conditions. However, but I think it is important to think about not sustainable food production, but sustainable soils, and how we can actually build and probably enhance microbial resilience in our soils. If you remember all the functions I was showing you at the start of the presentation, it's not only about food production, it's about all the other ecosystem services that the soil provides. But in other words, what I'm trying to say here is I think we now have the tools with molecular methods and so on to starting understanding what's happening in the soil microbial communities, how the different microbial groups change across the season. This is just one example from one of our experiments in here. You can see the different colors represent different bacteria groups, how they change across the season and change across different treatments and so on. And most importantly, which kind of effects are we actually creating? What we do this year in our soil, it will have an effect on the plants that might be growing next year as well. So understanding this full rotation system is very, very important in my opinion. So wrapping up and coming back to the start of my talk, we do now know quite a lot of what is going on inside of this previously referred black box. It's not that black any longer. And we do know now that to have a healthy soil, we need to consider the many several types of organisms that we find or not find in there. Plant-soil interactions are very complex, are quite challenging sometimes, and are very exciting in my opinion as well. I would like you to invite you all to think, if you haven't done it yet, about all the wonderful activities that are happening on our feet when we walk on soils. And with this, I would like to thank once again the British Mycological Society for giving me this opportunity to talk to you about a little bit of these incredible organisms. And thank you very much to you all for listening. I am very happy to take any questions or comments you might have. Thank you.
Okay, well, I firstly want to thank Sandra very much for a really informative talk. I think you were very clear in explaining the soil biology and what you led you into this particular field. And it's nice to hear someone talking about the whole system, looking at, two very diverse groups of organisms with fungi and cyanobacteria. A few questions have come in. Please do post more questions into the chat. I'm just going to kick off actually with one of the first ones because I think it's quite an important one that relates to maybe some of the commercial products that are available as well. And you mentioned that maybe their efficacy is lower than maybe in some lab experiments, and there might be issues there with the viability or even the content of those cultures. So Maxine asked right early on in the research, how were the fungi introduced to the soil and crops? How did you source them? How did you grow them? What kind of control measures do you have in place to set those up?
It's a very good question. So what we normally do is to try to knock down everything that is present in the soil by autoclaving the soil, for example, or steam treatment. And then we reintroduce the organisms that we want to put just to try to control what is going on. So normally you can buy inoculum and it comes in spores, hyphal fragments as well. When I grow my inoculum, you simply put it with autoclave sand and soil. Plantain is a very good plant to actually reproduce the fungus in there, and you create lots and lots of spores. These spores are then extracted from the soil, dispersed in water, and then you can apply a certain amount of spores, a known amount of spores, into your experiments. This is one of the easiest ways, time-consuming, but easiest way to do it.
And how do you source your mycorrhizal fungi in the first place?
Good question. So as working at the university, of course, there are several places that they can sell you inoculum for scientific studies, which then you know exactly the strain, the history of these strains, where is it coming from, in which kind of conditions it was isolated, and so on. But this is something you can do at home, of course, and you can just go and start extracting spores and seeing what you get. You will not know which species you have unless you use sequencing methods. But there are the two sources of inoculum, I would say, or asking colleagues, of course.
Of course. And actually, that was a question from me now, which relates to this, is that following your inoculation, do you follow the course of infection? Do you check, 1 month, 2 months, 3 months down the line, whether or not the inoculum that you initially provided is still there and is still active?
Depends. That's a very good point, of course. What I normally do is at the end of experiment, or maybe in the middle of the experiment, depending on what I'm looking at, I collect some root samples and you can stain them. One of the pictures I was showing, it was blue. You can stain the fungal hyphae and the fungal structures using trypan blue. And basically you look them under the microscope and you estimate the total length of root that is colonized by fungi, how many arbuscules are produced, and so on. You can use molecular methods, of course. You can extract the spores that are produced in the soil. Depends on the question a little bit, but yes, you need to check that you actually put an inoculum and that the inoculum does something to the plant and it's in there.
Okay, so there's a really important question that came in from Adrian, which again links into this, which is if the cyanobacteria are photosynthetic, then how does their ability to exist on the surface or subsurface of the soil impact on the interaction with the plant root system? Are you getting some partitioning of growth and their effect on the plants?
Not sure if I follow this one. So some of these cyanobacteria can live like free living organisms. They're not inside the roots. Some of them has been shown that they are actually inside the roots as well. Not all of them photosynthesize in that situation, I assume now. Make me
Okay.
think.
I guess the view is that cyanobacteria are blue-green algae. They will be dependent on light for growth. So is
Yeah,
that not always the case then?
I believe that some of them are inside the roots and probably there's not that much light. You really make me think with this one.
Okay, good
Thank you,
question.
Adrian.
Thank you. Thank you very much, Adrian, on that one. Okay, another question here. This is from Gary. Gary, first of all, commented upon the minor spelling mistake in one of your graphs where you replaced barley with barely. And I think that was just an indication that there was hardly any response there, very small response at all. But he also asked, what would your estimates be of the best yields per hectare in terms of scale up be from what you've seen in the best case scenarios? How big an effect is this actually going to have on agricultural production of foods?
So in my experiment, this is the one, and to apologize, that figure I show you, it was taken from a scientific paper, which is not mine, but sometimes there are spelling mistakes and things happen. In my experiments, I did so the width to the density that you usually would do in the field. I did not calculate, and this is a good point as well, how would that translate into the field condition? And I think it's important yet again, when we think about experiments we do in control conditions in a pot experiment, we try to mimic the natural environment, but it's not the same to grow plants in a pot. It doesn't matter how big the pot is compared to the field. They're much more, they're different, let's put it this way.
There's certainly a complexity in the
Mhm.
natural world that you lose from these lab experiments. Mike asked a question for the dual inoculation experiment. How was the wheat variety that you used
Mhm.
in the experiments chosen? Would different varieties perhaps have different responses?
Definitely. So the one I used was Skyscraper, I think it's called. And I basically chose it because I had it available. That was the reason, nothing else than that. I mean, it is used, at least around here as well. But yet again, we do know that the effects you see, it's very difficult to generalize. The effects you see will depend on the mycorrhizal fungus use, on the cyanobacteria strain, on the plant species, on the variety as well. So it is complex. And yes, variety would most probably have an impact.
Okay. Okay, this is a question from Gareth. He talks a little bit about commercial products. Are these products region specific? And actually, that kind of
Mm.
stimulates a question that I had, which was about potatoes. Um, Potatoes obviously are not native to the UK. They're
Mhm.
introduced, and I'm sure lots of other crops are introduced
Mhm.
as well. So those natural microbial communities are going to vary from region to region. Those kind of thoughts entered your research designs?
Definitely, definitely. And I think in this kind of globalized world, this is my personal opinion as well, it's quite important to understand what are we doing, not just moving plants and animals around, but also which kind of soil microbes are we moving around and what potential effects these soil microbes might have when introduced in the ecosystem. For the commercial products, and this is a bit of a disclaimer, none of this is my studies. Let me reiterate, some commercial products work in my experience, some work a bit less than promised, perhaps. There seems to be a bit of region-specific differences, which could be explained by many different reasons, I would say. Quality controls, different strains, different formulations even. This I am not sure. But thinking about the potential problems of introducing foreign strains is something to consider at least.
Yeah, I mean that's a point that also appears in the question as well about introducing foreign microbes to our native soils. There's a few questions here that kind of relate to the impact of these microbes on soil structure. Um, Again, I guess if you autoclave soil, that's going to significantly impact on the soil structure before you start. But have you looked at the actual soil structure?
Yet again, not in this experiment because we wanted to try to get out all this complexity of the natural wall by trying to control as many factors as we could. But as I was trying to mention, I think the important step is you can find something in the lab, but then you need to trial it in the field, and not even just for one season, but what is the effect of different seasons even.
Yeah,
Sorry,
I
the
mean,
question was
UK
about soil
soils.
structure. Yeah. So
Yeah,
they affect soil structure. Yet again, normally these experiments are relatively short term, but it's very interesting to measure more than soil structure, the amount of organic matter that is produced or being kept in the soil as well, and to understand all these physical, chemical, and biological properties of soil. So for me, the approach is to actually get the comprehensive view as much as you can. Everything is important.
okay. So David asked the question, what about hydroponic
Hmm.
production? Uh, is there any evidence that
Hmm.
adding arbuscular mycorrhizal fungi or cyanobacteria can impact on that? And a specific mention of adding them in rock wool.
This is something I must confess I have not come across. I do remember reading about some studies with mycorrhizal fungi in hydroponics. The Not the problem, but the thing with hydroponics, as far as I'm aware, because you control the amount of nutrients and everything you are putting, it doesn't make much more sense to put mycorrhizal fungi because the nutrients are already in the solution. I mean, they don't die in water, so potentially something could be applied. And I am not familiar with this rock pool, I must confess. But if it's some sort
I
of
think it's an
substrate,
inert substrate that
I don't see why not.
the builders use. I know that.
Right, then yeah, probably
I'm
something to
sure
look
somebody can put in the
for.
chat a little bit more about rock wool.
Hmm.
Simon's asked a very important question as well. And again, it's something that I was thinking about. The UK soils work extremely hard. Um, I live in a rural environment, and I see the rotation of the crops on an almost monthly basis. And fields never lie fallow for more than
Mhm.
days before something else happens. And I'm sure there's lots of planning that goes into that. But if the plants actually become even more efficient in extracting nutrients from the soil as a result of these manipulations, is there a risk that the soil actually will become depleted of nutrients even more quickly? Um, and we might end up in with a worse situation than we started off with.
Yes, it depends what you do with the soil and how do you manage the cultivations as well. I mean, that's why, in my opinion, cover crops, no tillage, for example. I'm not just thinking about mycorrhizal fungi, but try not to bother too much the soil community, I would say. If you are taking all the biomass of the plant, of the grain and so on, all the nutrients out of the system, at some point you will need to reintroduce the system. But that's where, for example, reintroducing the biomass that you don't use, for example, could be. I mean, I think an option to consider, absolutely.
Okay. Okay. If people want to keep on putting questions into the chat, that'd be really, really useful. I've got a really kind of philosophical question, which is about what you think fungi contribute to the global carbon economy.
Mm.
Um, are fungi as a whole net producers of CO2 as they are decomposers? Or are they, as a result of their mycorrhizal associations, tipping the balance the other way.
We're talking about fungi or arbuscular mycorrhizal fungi.
Well, fungi as a whole.
As a whole, I have no idea, I would say, if you take into account all the decomposers and everything. I do remember some relatively recent studies from some colleagues and friends of mine that they estimated the amount of carbon that is actually locked in the mycorrhizal hyphae and structures as well, and it's quite considerable. That's why I'm saying just arbuscular mycorrhizal fungi, there has been some measurements. Ectomycorrhizal fungi, something of course a bit similar but different. There is also a lot of carbon in the soil that is locked in there, which is good. Fungi, the whole fungal kingdom, I am not sure what to say, I must say.
Obviously, as the British Mycological Society, we're always
Mm,
looking to answer that question. It's one that the public do come up with on a regular basis, and I must admit that I'm somewhat stumped by that.
mm.
We want to promote fungi as being on the good side, but
I think they're on the good side, and even though, I mean, I'm thinking decomposers, yes, they produce CO2, but they need to decompose material for the nutrient cycling. So this is also something I was trying to put in my talk. At the end of the day, there is not bad organisms. They all have a function. It might not be the function you want if you have a crop,
yeah,
but they have a reason for existing, and they have a place in the trophic web, for example. So this is where I struggle a little bit. I think everything is almost needed in the right amount as well, which is more challenging perhaps. But all fungi are good, absolutely.
yeah. Some of your graphs did show a negative effect on plant growth. Obviously, the percentages were generally on the positive side or zero side. Um, but I mean, are you aware of specific species-specific interactions which really can tip the balance between pathogenic? I mean, I look at arbuscules uh, and I look at Horstoria because I teach about these things,
Hmm.
and to me they look pretty much the same.
Hmm.
I mean, have you got any thoughts on the relationships between those different structures and that?
Same idea, absolutely same idea. I mean, some fungi became pathogenic and some fungi became a bit more clever, in my opinion, and became mycorrhizal, which is a win-win situation.
Okay. So linking to that, Gareth has asked how much do we know about the cell signaling
Mm.
involved in this symbiosis, and what kind of methods might you use to identify suitable organisms for fruit crops? They might be two different questions.
We do know quite a lot of what is going on, and especially this molecular communication. So we do know that the fungal spores, arbuscular mycorrhizal fungal spores, are sensing the environment. We know of some factors that are actually telling the spore, now it's a good time to germinate, because of course you are depending on finding a suitable host. So you really need to make sure that when you germinate, there is a potential root next to you. So they have this signaling is understood in the sense that they know how to get awake and germinate. Some plant species, yet again, they do sense, and I'm putting everything in the sense way, of course, chemical communication, where there is a fungal inoculum potentially good for them as well, and they send more signals as well. And from very early on, we do know quite a lot about this communication. It's quite complex, of course. I believe, yet again, thinking about how the carbon gets into the fungus or how the phosphorus gets from the soil to the plant, all these transporters, all this communication in the cell is becoming to be more and more understood. But as far as I'm aware, we don't know of all the transporters. And we do know that things move, but we don't know how. And I think this is quite fascinating yet again, because
Mm.
biotechnology, that could be something to encourage perhaps as well. I make it sound like it's very simple. This is not my field, but it's very interesting.
Okay. I work in the medical mycology field, and obviously we're interested in patient resistance to infections.
Mm-hmm.
Plants have an immune system.
Mm-hmm.
They show systemic immunity following colonization by
Mm-hmm.
some fungi. Do you have any insight into whether or not these mycorrhizal associations are actually kind of beneficial in terms of promoting resistance to fungal infection?
Yes, yes, potentially. And yet again, once the plant is colonized by mycorrhizal fungi, there is a lot of different metabolic processes happening, an increase in secondary metabolites. I mean, it's not simply because they get more nutrients. There is a lot of genetic changes, molecular changes there. Some plants become more resistant to things. Yet again, I guess it depends a bit like humans, our immune system. So the plants allow the mycorrhizal to colonize. They don't kill it in that sense because it's good, and the fungus has knocked down these defense mechanisms as well. But plants are not stupid. Fungi are not stupid, but plants are not stupid either, I must confess. And I didn't mention that, but it's not a plant root system may have 10 different species of arbuscular mycorrhizal fungi happening in the same root as well. There is a lot of competition for space from the different fungus as well. So we do know some things, how this competition is created, for example, whether you are colonized by one fungus A, then makes it easier for fungus B to colonize or not. But the exact mechanisms, yet again, is quite obscure at the moment.
I think the notion of mycorrhizal fungi having to suppress the host immune system in some way so that they can actually establish this relationship sounds interesting. I don't know if mycorrhizal fungi produce typical effectors, for example, that in common with plant pathogens that are trying to avoid host immunity. Certainly sounds like an area for exploration if it isn't
Absolutely.
already. Adrian's asked, is quorum sensing involved in the interaction of the fungi for root colonization? And I guess that could be applied both in the context of just mycorrhizal fungi interacting with the plant, but also the cyanobacterial fungal interaction.
As far as I'm aware, absolutely. And yet again, there is a lot of communication, chemical communication happening, going on. It's not my field yet again, but I have done some experiments with my students looking at bacteria communication, bacteria-bacteria communication as well, and how they can react to specific amino acids, for example. Plant system, plant roots, they exudate a lot and lot of compounds. We have a lot of soil organisms. It's not just fungi and cyanobacteria. It's tardigrades, it's earthworms, it's herbivores, it's lots of different things producing lots of signals as well. So it is, yet again, challenging and complicated to understand because we tend to simplify the experiments to understand what is going on. But yeah, absolutely, there is a secret language underneath our feet.
Okay. I can't see many other questions in the chat at the moment. There's one from Nasima, though, which might be a good wrap up, which is, nowadays, are there any approved products that are based on both a mycorrhizal and a cyanobacterial combination?
Good
Is that
question.
is that your plan?
I don't have a plan. I always tell, I'm not here for the money in academia. I'm here for the finding the answers to some questions. I'm not sure. I am not sure. I must confess I have not looked at it, but potentially could be a business idea for somebody.
I'm sure your university were very happy to hear that the
Always.
potential is there. Tim's actually just posted a question. Is there a way of assessing soil micropopulations to determine whether inoculation is worth a try? And I think you did present some uh, microbiome studies at the end of your talk. And he says that the data suggests that if the flora is rich, it's not worth adding to it.
Um, Good question.
Question mark.
That's a good question. There is ways to assess microbial populations, of course. If you have money, next generation sequencing, you just, I mean, it's not as simple as I make it sound, but you just extract the DNA from the soil organisms that you are interested in. You just use primers and you can have some kind of profiles of the microbial communities. This is just showing you presence absence of something. And yet again, if you take a soil sample, I might find something here, but if I move 20 centimeters, it might not be there as well. So this kind of sampling effort is quite important. Across the season as well. Um, it's good to know whether you have pathogens. And of course, this is something that more applied farmers are very interested to know whether they have a pathogen in their soil. Worth trying. If you have a lot of money, why not? It would be very interesting to understand what's going on, I would say.
Okay. So last question for me then is what's next?
What's next? Good question. Publish this study because it's on my desk. And basically, I'm continuing with these net zero projects and trying to more and more understand what is happening in the soil.
Okay. Well, I'd like to thank Sandra very much for this evening's talk and entertainment, because that's an important element of these presentations as well. I think we should wrap it up just to say that there are lots and lots of really positive comments about your talk this evening. So thank you very much, Sandra. And I think we'll bring this meeting to a close. So thank you.
You're very welcome. Thank you, everybody. Thank you.